Hello everyone and welcome. I'm the host of today's meeting, Jessica Tu, from GenScript ProBio Marketing Department. On behalf of GenScript ProBio, I'd like to thank you all for attending today's webinar. GenScript ProBio is a subsidiary of GenScript Biotech Corporation, proactively providing end-to-end CDMO service from the drug discovery to commercialization with proactive strategies, professional solutions, and efficient processes in cell and gene therapy, vaccine, biologics, discovery, and antibody protein drug to accelerate drug development for customers. This webinar will include two talks. One talk will take 30-minute presentation and 15-minute Q&A session. You can submit questions at any point during the webinar using the chat or Q&A panel situated in the menu on the right. Now, I'm delighted to introduce today's first speaker, Dr. Xiao Pan, Director of Platform R&D Department, manages the Platform Process R&D Department to develop process R&D platforms based on antibodies, proteins, and gene cell therapy. Dr. Xiao Pan received his PhD from the Department of Bioengineering, Wageningen University, the Netherlands. In 2017, he worked as an important scientist at Fujifilm Diosynth Biotechnology Company in the United Kingdom. As the technical director of the Upstream Cell Line Development Department and Process Development Department, including process design, development, amplification, and characterization. Dr. Pan joined GenScript in 2018, responsible for the upstream process development process, and led the CMC work of more than 20 R&D projects. During today's webinar, Dr. Pan will discuss integrated mRNA one-stop solution. Now, I'm delighted to hand over to today's speaker, Dr. Pan.
Hi, thanks host for introduction. Can you hear me well?
Yeah.
Okay, I'll share my presentation.
Okay, please.
Can you see my screen now?
Yeah, it's great. You can start.
All right. Well, hi, good day, ladies and gentlemen. Thank you for attending today's webinar. My name is Xiao Pan. I'm the head of Platform R&D Department and mRNA business in GenScript ProBio. Today, I'll be giving you a presentation with the title Integrated Solution for mRNA Vaccine and Therapeutics. To start with, I want to share with you a brief introduction to our GenScript group company. We are a group company with 4 sister companies, which are GenScript, known for its life science service and products. And we have BestSign, dedicated in providing and producing industrial synthetic biology products. We have Legend Biotech, which is focusing on cell therapy and deliver drug to the patient. And we are a GenScript ProBio. We are the GenScript CDMO sector. And we focus, as previously our host mentioned, we focus on the biologic CDMO and the cell and gene therapy CDMO. This is the content of my talk today. I'll start by giving you a brief introduction to our ProBio solution to mRNA vaccine and therapeutics. This slide shows a brief history of our business footprint in the cell and gene therapy field. It's been always encouraging to see that we have assisted our customers to obtain the first CAR-T, TCR-T, mRNA, and CRISPR gene editing IND filings in NMPA in China. And for mRNA, we are pretty young. We started the business since 2021 from IVT to LNP encapsulation. We officially launched the service, and up to now, we have completed over 20 IVT-mRNA and LNP GMP batches and we delivered all the projects with 100% success rate. At the moment, we have 2 projects that are at the filing stage of NMPA and US FDA respectively. So this is a bird view of our Plasmid and mRNA facility, which are based in Zhenjiang, China. The facilities are designed to very flexible and to avoid cross contaminations, to ensure our fast delivery of the products. As you can see here, we have Plasmid facility ranging from, with a production scale ranging from 5 liter to 500 liter. And we have mRNA facility covering process development, GMP manufacturing, all the way till drug product fill and finish. Sorry. From research to commercialization of mRNA drug development, we offer phase-appropriate product grades and quality controls for each respective products. And from plasmid to linear plasmid to IVT and LNP mRNA, we have different product offerings focusing on the different stages of the product life cycle. With this, we can effectively produce the material with a very economical cost and timeline. For mRNA drug development, leveraging our GenScript group capability, we provide one-stop service all the way from gene synthesis to mRNA LNP GMP production. Here we compared our traditional approach, which is separated plasmid and mRNA CMC, for instance, in different CDMOs, or our customers do the plasmid with ProBio and do the mRNA by themselves. But now we have integrated these 2 services into a complete plasmid and mRNA service. Putting them under one roof shortens the timeline from the previous 10-month time to the current 6 months time. So within 6 months time, we can go all the way from sequence to the first GMP DP drug product ready. So in the next few slides, I'll give you some introduction to our CMC, to the mRNA CMC challenges and our strategies towards it. To borrow some figures from Nature reviews on the mRNA application, as you can see, the layout of the application field for mRNA is very broad. It includes preventative vaccines, cancer vaccines, tumor immune-related therapeutics, regenerative therapies, autoimmune and secretory-related therapies, and so on and so forth. Up to the recent data, majority of the mRNA pipelines are related to mainly the infectious disease and cancer treatments. The mRNA application is boosting so fast due to the fact that it is safe with a clear structure to activity relationship. It is also safe compared to DNA and the virus-based delivery, since it's non-infective and is non-integrating. It is also very versatile in terms of the protein expression, so it can code various of different proteins And the delivery of mRNA using LNP enables various, for instance, administration routes or organ or specific cell targeting approaches. And what's more important is that the mRNA manufacturing is friendly and for its mass production, and is relatively easy to be scaled up. In this slide, we have shared with you the CMC challenges that we consider the most important for the drug development. So for mRNA, we're focusing on the stability, efficiency, immunogenicity, safety, and effectiveness. We have to combine these considerations and take them as a whole to look at the CMC development. And to start with, mRNA sequence design, and process development, and design space exploration, all the way until the mRNA delivery. These are three key impacting factors to address these mRNA CMC challenges. And these three parts are also what ProBio mRNA platform has been focusing on. We spend a lot of time doing optimized sequence for customers to develop a very robust and scalable process. And also, we have recently done quite a lot of successful gram scale up of LNP manufacturing processes. So until now, we have established the integrated mRNA manufacturing process, including 4 platforms, ranging from the circular plasmid to linearized plasmid and to IVT-mRNA, where we can do both traditional enzymatic capping and co-transcriptional capping. Then downstream purification for these IVT-mRNA to formulate the drug substance. And then this drug substance can be LNP encapsulated to formulate the mRNA drug product. In the next few slides, I'll zoom into our platform, technical platform, to give you some more detailed picture of how these platforms look like, with a focus on the IVT of mRNA and LNP platform. For the IVT process platform, we have accomplished more than 20 batches of GMP production with 100% of success delivery. We have IVT-mRNA process platforms covering 3 different types or formats of mRNA, namely the regular mRNA, linearized regular mRNA. We have the self-amplifying RNA and the CircRNA. We have a very well-developed process for the regular mRNA, characterized with high production yield for both co-transcriptional capping and enzymatic capping methods. Together with the high purity and capping efficiency, we have also obtained very good product yield and recovery rate. The self-amplifying RNA, or so-called saRNA, is known to have extended expression in vivo, and we have tested up to 12 kilo base pair length of saRNA. The process results in very high purity with very good production yield. The good thing for CircRNA is that it is stability in vivo, and in addition, there's no capping and tailing processes needed for CircRNA, meaning that the raw material cost during the manufacturing is significantly lower compared to the other 2. But the downstream processes to remove the non-circularized and the nicked RNA during this process is more challenging in the industry. And in ProBio, we have our proprietary purification processes to address these issues. And the purity we achieved out of this process is typically greater than 90%, which is a very good number. And above them all, we have the dual sourcing system for all three different structure mRNA manufacturing. We have our China domestic raw material supply option, as well as the international supply for all the raw materials. And these three different processes we ensure the GMP scalability and that are compatible with further scale up and commercial feasible manufacturing. This page shows just gives you a brief introduction on different capping methods. As probably many of you have already known that the enzymatic capping, such as vaccinia capping enzyme, is used in the enzymatic capping process to convert the 5-triphosphate group to the cap0 structure. And then the cap0 can be modified to the cap1 structure by the 2'O methyltransferase. Apart from it, the enzymatic capping, we're also partnered with TriLink to provide the CleanCap co-transcriptional capping process. For this process, the cap analog is the cap analog, the CleanCap reagent is introduced to the process directly during the IVT step, where a competitive complementation capping process happens, resulted in high capping efficiency of the analog onto the target reagent directly. So there are some data here we show here to give you some real world cases on our IVT-mRNA platform. We have done a lot of exploration in the capping process design space, using design of experiments to explore a wide range of process conditions, and then increase the process parameter granularity to find the key rate limiting steps. And finally, we logged the process, and then generally they gave a very good scale up process. In this study, we have improved our recovery rate during the process development and gradually scale it up and lock the process with a very good recovery rate. In this specific case, we increased the capping efficiency for two product molecules from 40% to 90% in the end, and we achieved consistent yield and recovery rate during the process locking and scale up. And these are all done with the help of design of experiments to identify what is the problem during the initial low capping process. In this slide, you can see that our platform process is compatible with different mRNA length, ranging from 1.2 to 3.5 kilobase pairs. And we compare two capping methods. We both have very good capping efficiencies. And to the right, you can see that we are accumulating the stability data of the large-scale mRNA products. And overall, the product stability is very good under different conditions. And we are keep growing this stability data and expect to achieve, for instance, 1 year or even 2 years stability data. After the in vitro transcription of the mRNA, downstream process steps were done to further remove the process and product-related impurities. Typically, the one-step affinity chromatography is enough to guarantee the high purity of the mRNA. But in some specific cases, where we want to remove the double-strand RNA or truncated RNA, then a polishing step can be introduced to the process. And overall, we have a downstream process with a recovery rate ranging from 50 to 80% for the whole process. And strong analytics platform is another key to success. Later on, Dr. Lauren Lu will also give you more detailed picture of ProBio quality control departments. Here, I want to stress that leveraging our strong capability in gene synthesis and the nucleic acid drug development, and we have established quite a very strong AD and QC platform, where we have orthogonal release methods for the mRNA different product attributes. For instance, the CE and HPLC and gel electrophoresis to look at the purity of mRNA from the different angles. And also we have accurate high resolution LC-mass-based methods for capping characterization and poly-A characterization. In the next few slides, apart from the previous mentioned the regular mRNA, we also offer very high performance saRNA and CircRNA platform. In this saRNA platform, we managed to improve the purity of saRNA with a length over 11 kilobase pair. From the originally around 60% of purity to over 95% of purity. And this process was with good product yield and recovery rate. A lot of DOE were done to make this happen. And then we have also demonstrated that the produced self-amplifying RNA has demonstrated expected strong and long-term activity in vitro. For CircRNA platform, we can support various ligation methods for our customers. For example, the intron self-splicing methods or the T4 RNase ligation methods, we have both process and a technical platform to support. And I have also incorporated a brief manufacturing process flow for your information here. And we designed several CircRNA ligation sequences that are shown to have good circularization results. Here you can see that the sequence results show our ligation site is complete and very accurate. What's more, with a proprietary circular RNA purification process, we managed to effectively remove the impurities with, for example, the non-circularized mRNA and the nicked RNA during the downstream process, resulting in very high mRNA, circular RNA purity analyzed by CE. So in the next section, I will focus on the lipid nanoparticle or formulation development process platform. Here, our keywords are that we have accomplished gram-level LNP encapsulation GMP production with very good release results. And we have been focusing on stringent control of the morphology and the functionality of the developed and produced lipid nanoparticles. First, I want to iterate the importance of LNP to mRNA therapeutics. mRNA needs delivery vehicles to protect it from the nuclease in physiological fluids to evade the interception and clearance in the body. And after reaching the target cells, it needs to be internalized by the cells and escape from the endosomal to exert the translation. And there's quite a long way for the mRNA going to function. And that's why we have spent a lot of efforts on the mRNA encapsulation and quality control for different formulation development programs. ProBio have established microfluidics-based LNP process platform. From process development up to GMP production scales, where we are also developing a T mixer or a so-called jet mixer based processes. But the equipment all these equipment have built-in functions to keep similar flow dynamics and mixing for the LNP encapsulation, which ensure the consistent scale-up of our LNP processes. And for our LNP platform, we can produce from several milligrams up to 6 grams of mRNA in different grades. So here the pro grade refers to the research use grade, and the GMP grade is done under full GMP conditions. We are also very flexible in the LNP formulation. We can customize it for you. We can develop a commercial-based LNP formulation for specific projects as well. This table details the work scope we provide during the LNP formulation development and production. I will not go into the details of this table. It is quite a comprehensive development scope covering formulation development to process development to quality study and the manufacturing scale up. And within each of the service stage, we have a carefully selected work content based on our platform knowledge. There are rounds of design of experiments for the process and the formulation development to be able to lock the process and to ensure a robust process scale up. So this, as you know, that the size, the morphology, and the physiochemical characteristics of the LNPs are very important, that they determine the mRNA biodistribution and cellular uptake. And thus, they determine the efficiency and efficacy of the mRNA. These characteristics of the LNP can be optimized through formulation optimization and process optimization. For example, flow rate and mixing parameters, things like this. The LNP platform that we have is quite universal. We have tested multiple LNP commercial and proprietary formulations on our platform, and the physiochemical characteristics of these LNPs are all all satisfying and very consistent. So for during the formulation development, we can also assist our customer to optimize their specific LNP formulation based on the specific application scenarios. For example, is in vitro or in vivo. Here in the left case, through lipid composition design of experiments, we were able to identify the formulation specific for certain cell types, but not for the other cell types. And what is more interesting is that a certain LNP formulation is only functional in, for example, in this cell type B, but not in the other 3 cell types. So this is a demonstration that we can control the functionality of the mRNA delivery using LNP by the formulation development, through formulation development. In this case, through the optimization of the commercial formulation, we found better conditions resulted in extended mRNA expression in vivo compared to the original commercial formulation. As you can see that this blue line represents the original commercial formulation, and we optimized the formulation, and then it shows better mRNA activity along the time compared to the original formulation. So there's a great potential even in the commercial available formulation if we design it well for.
Hi, Dr. Pan, can you hear me? We can't hear Dr. Pan now. Wait a minute, then let me contact her.
Hello, can you hear me?
Yeah, it's okay now. Yeah, you can reshare your PowerPoint.
Okay. Can you see my screen now?
Not yet. Wait a moment. It's okay now. Yeah, please.
It's okay now. Oh,
Yeah.
sorry for the interruption. There are some technical problems here. So it's all good. Okay. I'll continue. Okay.
Yeah, please.
Right. So for I'll start over this present for that. For mRNA characterization, we know that morphology and nanostructure are very critical to the functionality of the LNP. Not only the formulation and the percentage of lipid composition, but also we should zoom into the details of the morphology of the lipid nanoparticles. Here we have them. It is known that the formulation itself and the encapsulation process can both impact the LNP nanostructure. There with different formulations, you can see very drastically different mRNA morphology, and even with different pH conditions, buffer conditions, or different mixing conditions, the mRNA detailed structure appear to be quite different, which will have a following impact on the in vivo or in vitro release of the mRNA. So in ProBio, we have pay extra attention to control the LNP nanostructure, and through that to control the LNP functions. We have done thousands of cryogenic transmission electron microscopy studies to link our formulation and process conditions with the nanostructure performances. So you can see that with the quality by design concept in mind, we're connecting the process input parameters to the nanostructure and eventually to the mRNA-LNP safety and functionality. This slide the main point is to show our GMP capability and capacity. We have completed more than 10 batches of gram scale GMP production for the lipid nanoparticles, and we have run all these batches by complete process control and flow ranging from encapsulation, formulation to sterile filtration and fill and finish. And we can encapsulate 2 to 6 grams of mRNA within 3 hours. And the DP concentration we've tested ranges from 0.1 to 2 milligram per ml, which is a quite high drug product concentration for the mRNA. And what's more, we can do both liquid formulation as well as lyophilized formulation program. And in this figure, you can see that we have completed in real world a gram scale production with a very good particle results with a size around 17 nanometer, PDI 0.1, yield around 80%, and final encapsulation efficiency around 95%. So to summarize, here we have established a very well-developed LNP platform with a wide range of mRNA applications. And we are open in formulation development service. It can be commercially available formulation. It can be your customer's own lipids or formulation. It can also be a co-development model. We can discuss that if you are interested. We have tested multiple payloads and they all shown to be working in vitro and in vivo. not only mRNA, we have saRNA, CircRNA. We have also tried to encapsulate DNA and sgRNA. And we have scaled up scale up capability to up to 6 grams of mRNA per batch, DP concentration up to 2 milligrams per milliliter. ProBio has also quite unique pharmacology group that supports our process development as well as GMP release. We have excellent physiological and morphological characteristics during our development, and we have integrated in vitro and in vivo testing that are available to confirm our formulation development processes. So in the end, I believe this is one of my last pages. Here I want to iterate that we have our Plasmid mRNA platform is a technology and application scenario driven, and we have set clear goals that for each respective application scenarios, whether it's in vivo, in vitro, it's a vaccine or cancer vaccine, or even a personalized cancer vaccine, we all have a representative or respective service solutions for those programs. And we have within all the programs that we did for the mRNA, we have obtained more than 10 IND approval from FDA, NMPA, PMDA, MFDS, and other authority agencies, which are with all these track record listed in this table. And for the IVT-mRNA, we have tested both enzymatic and co-transcriptional capping, resulting in good capping efficiency, a good recovery and production yield. We have also iconic self-amplifying RNA and CircRNA platform that are very well performing. For the LNP, we have tested a wide range of different LNP formulations and the lipid raw materials, and they are all behaving very good on our platform, with encapsulation efficiency higher than 90% in our GMP productions. We have also established internal capability to characterize the LNP nanostructure and morphology very well. And we have very stringent control input parameters to control this nanostructure of the produced LNP. So yeah, with that, I want to conclude my presentation, and thank you for your listening. If there are any questions, please feel free to let me know. Thank you.
Thanks for Dr. Pan's excellent presentation. If you have any questions, please type it on the chat or Q&A panel. As you enter questions, please also follow our LinkedIn account, GenScript ProBio, and Korean audience can also scan this QR code and follow our Kakao channel. There will be more online resources, and we will share them on our LinkedIn and Kakao channel at the first time. Okay, we received some questions from the audience. Let's take a look at the first question. Hi, Dr. Pan. When entrusting CMO, I am curious about the minimum mRNA production of cGMP grade. Could you please answer this question?
Yeah. Yeah. Thanks, host, for the question, and thank you for asking. So this question relates to the production scale or quantity of the products for the cGMP grade mRNA production. I think it's so first, ProBio is pretty flexible in terms of the production scale. As you can see during my presentation, that we have for the mRNA, we have several hundred milligram up to gram scale production. And for LNP, we have the same, we have similar from a couple of hundred of milligram up to 6 gram of LNP encapsulated mRNA production. So in terms of the minimum scale, I would say it's around 100 or 200 milligram of mRNA per batch, because below that, your process scale is too small. That probably is not very representative of the commercial production. So even if the GMP material that you need is very small amount, we suggest you to use relatively more representative scale to produce your mRNA, because other than the mRNA material you need for your clinical study or for some IIT study, you also need materials for stability study, for your analytical release study and things like this. And of course, you also want some retains to compare to your future samples in the next stage. So yeah, a quick answer to that, I would say 100 milligram to 100 or 200 milligram, that would be the minimum mRNA GMP production scale.
Thanks for Dr. Pan's answer. Let's move to the next question. My LNP was happen aggregation in free sol cycles. Why this happening?
So the LNP is aggregated during the free sol cycle. That's I think that's a pretty common phenomena or results that we obtain during our process development as well. Because during the free sol, the crystallization of the water or buffer can jeopardize the LNP nanostructure. So it's not surprising to hear that the LNP aggregates or it loses its functionality during the free sol cycle, and we happen to encounter that a lot of times. And so in that case, we also have solutions, for example, to add protective reagents during the buffer solution or the formulation solution to protect the lipid nanoparticles from the free sol activities. Yeah.
Thank you for your clarification and solution sharing. Yeah, and the next question is, based on the development so far, how do you compare mRNA and CRNA in drug development?
So if I understand this question correctly, the CRNA is referring to the circular RNA. So compare between mRNA and the circular RNA, I think they, yeah, it really depends on the application scenario of the specific drug. So it's really highly correlated with the MOA of the drug. Circular RNA is known to its stability and quite a stable structure for especially in vivo, so that the expression longevity is typically longer for the circular RNA compared to the mRNA. We also have internal data to support that. For example, mRNA with the expression level maybe 3-4 days, and the circular RNA can be extended up to over a week or even 2 or 3 weeks. Well, 2 weeks, maybe not as long as 3 weeks. But the manufacturing of the circular RNA, as I mentioned in my presentation, is more challenging. You have to there are more product-related impurities to be removed. And these are, you have to also have a very good analytical methods to identify these impurities, because many impurities are very similar to the circRNA itself. So by just size or CEX chromatography, you're not able to really distinguish the mRNA and the circRNA and its impurities. So there are challenges there, but I would think that circRNA is the future of mRNA because the nature of it, by nature more stable than the mRNA. So I would expect a growth, significant growth of circRNA processes or products in the future.
Okay, thank you. And the next question is, I want to know linearized pDNA purification method.
Yeah, I think as this presentation is focused on the mRNA, I do not prepare the material or process for the plasmid part. I would say it's if you have specific questions, I would suggest you to contact our ProBio BD representative for the DNA production. So I think I'm not really the expert of the plasmid, or I can divert your question to our experts if I have your contact. So please feel free to contact us.
Okay, thank you. And the next question is, when ordering mRNA, inquire about time and cost according to the production volume. Yeah, I guess maybe this question is better concerned about our BD. Yeah, but if you know more about the details, you can share too.
Yeah, I think it's a good suggestion to keep in contact with our BD, or just leave us a message and let us to approach you or address your question better. Oh, okay. I see a question on the.
Yeah, there is also a new question in the chat room.
So there's a question, does circular mRNA immunogenicity differs from linear mRNA? Yeah, I think it really depends on how you see the issue. As I mentioned, for the circular RNA, there are many by nature there are many impurities in the process, and these impurities are all the stimulators of the immunogenicity. So if you really have to compare them apple to apple, you have exactly the same sequence, and you have the circular RNA with, for example, 95% of purity, and you have mRNA with 95% of purity. If you put them together, I would believe that they are pretty comparable, because there's no way why the one has more immunogenicity than the others. But from the CMC perspective, we have to demonstrate that both two are the impurities are good. Will not mislead us in terms of the fair comparison. And the other thing is that there are direct answer to this is that we do not do the apple-to-apple comparison for these two mRNA yet. But for the linear mRNA, we have done a lot of efforts to measure the immunogenicity of the linear mRNA, and we try to modify the mRNA using different NTPs, and we try to remove the dsRNA, and it appears that these all contribute significantly to the immunogenicity of the mRNA. So I would not think it's just a comparison between the circular and linear RNA. It's more of for each respective RNA, we need to understand what is the causal effect of the immunogenicity, and we have to control it right from the very beginning, from the plasmid and from the process input parameters.
Okay, here is also a new question from the chat room. After DNA mass treatment to erase DNA template for IVT, is there any step to erase high salt, such as magnesium, necessary for the next downstream process? If you do you use DF or any dilution step, and how long does it take between IVT and column injection? If you don't do, I also want to ask the time between IVT and column injection.
Sorry,
Yeah, I also send this question through Teams. Yeah, you can check there.
Yeah, these questions are quite detailed questions. I think it would be better if we have if we can have a contact of this person, and then we can share some of our technical details to this, because I think yeah, I think just contact us, please, and we will share you some of these details, because I don't have all the questions in my head. I don't have all these answers in my head yet.
Okay, no problem. And the next question is, how could you distinguish between the NIC form and the CircRNA?
Yeah, we have, so first is that we have e-gel is another gel electrophoresis methods that are very often published on the papers. If you zoom into the papers, you will see a lot. So there are typically a band of your circular RNA, a band of linear RNA, and a band of nicked RNA in between. So that's the more it's a very straightforward analytical methods to identify the nicked form. And so if you we have tested many other conditions in for instance in HPLC and CE, it's not very how to say easy to distinguish compared to the e-gel. So currently we're using that methods to distinguish these two nicked and circular RNA.
Okay, thank you. That's all the question we have for now. Thanks for Dr. Pan's response.
Okay, thank you.